Welcome to Grand Rounds. Thank you for joining us today. We have a couple announcements. First, next week for Grand Rounds, we will have Dr. Dennis Mitchell, Executive Vice President for University Life, the Senior Vice Provost for Faculty Advancement, and a Professor of Dental Medicine here at Columbia University Irving Medical Center. His talk will be titled Faculty Advancement at Columbia University. Well, we have two reminders. First, as a reminder, we will prioritize questions from trainees during the discussion session after today's talk. We encourage everyone to post their questions at any time during the talk. You don't have to wait till the end of the talk, using the Q&A feature at the bottom of the Zoom window, which is separate from the chat. If you are a trainee, please put the word trainee at the beginning of your question. The second reminder is that we have begun compiling a list of potential speakers for the upcoming academic year and ask for your input. We say this every week, but we're really looking for new and interesting ideas. So please submit a nomination if you have one. We aim to have a diverse list of speakers that cover basic to clinical topics. If you can fill out our survey, Simon is going to post a link in the chat here as well with at least one nomination, or pass along the link to members of your group. We would greatly appreciate it. This link is going to be permanent and constantly monitored during the upcoming year. So if you forget this week and want to do it next week, it will always be there. Please continue to add names for future dates as they come up. With the co-directors, Jeff Miller and Kate Elkington, we value the input of our department members and thank you all in advance. For today, I would like to welcome Dr. Jay Gingrich. Dr. Gingrich is the Sackler Institute Professor of Developmental Psychology and Psychiatry at CUIMC. He received a BS from the University of Kansas, where he majored in chemistry. He then received both an MD and a PhD from Duke University. Notably, he did his PhD with Dr. Mark Carone, and his thesis was titled Purification and Molecular Cloning of the Gene for a D1 Dopamine Receptor from the CNS. He came to Columbia and NISP following his time at Duke. Here, he did his residency and postdoctoral fellowship with Dr. Renee Henn. And then in 1999, he became an assistant professor of clinical psychiatry and has been here ever since. He has been honored with numerous awards. There are too many to list today. They include the NIMH Career Development Award, the AFSP Award, the NIMH Axelrod Young Investigator Award, and the Connie Lieber Outstanding Research Award. He has received numerous grants from agencies such as the NIMH, NIDA, Simons Foundation, and Whitehall Foundation. He has well over 70 publications. Dr. Gingrich and his lab use a systems approach to better understand normal and abnormal brain function, in particular the mechanisms that underlie neuropsychiatric disorders such as depression and anxiety. Their goal is to understand how environmental, genetic, or epigenetic factors affect behavior and the intervening systems such as circuitry, anatomy, and physiology. His lab often exploits the ability to genetically modify mice to either mimic known human susceptibility factors or to use conditional gene modifications to further investigate hypotheses regarding circuitry and physiology. Jay is also pursuing several lines of research related to the role of serotonin signaling in the cortex, and these studies have demonstrated an important role for cortical influence on behaviors related to schizophrenia and anxiety. On a personal note, I have known Jay for nearly 20 years, and in that time, I have come to know a really remarkable person. Not only is Jay just a wonderful scientist, colleague, and clinician, he is also an expert in home construction. So if you have questions and you're doing it over your house, you go to Jay. It is just an honor to moderate this Grand Rounds today. The title of his talk is Revisiting the Safety of SSRI Use During Pregnancy. Are we causing more harm than good? Please again join me in giving Dr. Gingrich a warm welcome, and thank you so much for being here. I really appreciate it.
Thank you, Christine. Let me see. Am I sharing the right desktop, Simon?
Just switch the displays.
It's good?
Switch displays.
Oh, switch displays. Is that better?
Perfect.
Okay. So yeah, no, thank you, Christine. It's always a pleasure to give grand rounds at your own institution. It's I can't believe it's already been 20 years. Um, yeah, I must be a PGY. I don't know what I want to think. At least PGY 20. For the trainees, this will be something I'll try and explain as I go along, but this is really an interesting path going from, as you'll see, from some basic science findings integrated with some of my clinical thinking, and it's really led me in a somewhat unexpected direction. But before I got started, I just wanted to show some of the people who have participated and really were fantastic colleagues during the work that I'm going to present. Mark Ansorg has been a partner in the basic sciences from the beginning. Recently, Sarah Canetta has joined our division, and she has also pitched in. I have some work that I'm going to show of hers towards the end. And then on the epidemiology side, which we'll get to later in the talk, Myrna Weissman, Adi Talati, Alan Brown, Andre Sarander, and Ricky Wesseldorf have been fantastic colleagues, both here at Columbia and around the world. And then we've got some newborn imaging that I'm only going to show one slide today, but that was the collaborative effort of Jonathan Posner and Claudia Wigo, and then Brad Peterson, who's since moved on. And then also with the EEG and the recruitment of infants in our Conti study, Bill Pfeiffer, Michael Myers, and Catherine Monk were of tremendous help. The funding for the work I'm showing came from an NIMH Conti Center, several NIMH R01s, as well as funding from the Sackler Institute. So as the title, and I apologize for the provocative title, or maybe I'm sorry, not sorry, but it's been something I've been thinking about for quite a while, which is depression during and after pregnancy. And we've known for a long time that that depression in and around pregnancy is a big clinical problem. But the thing that I've been wondering about, actually from the time I was a resident, was we're clearly helping the mother, but what about the future fetus? Are we helping the outcomes of the second part of the dyad by treating the mother? And we always have, I think, hoped so, and that was, I think, the belief from the beginning, is that this would be a win-win situation. But I think it's important to revisit that. And the other part, I will take you through a not completely deep dive into what caused me to rethink this risk-benefit equation regarding the child, the developing infant during pregnancy, from what we learned with some of the basic science work going on in my laboratory years ago. I'm going to talk about some of our recent work suggesting a mechanism that may explain some of the things that we found in mice and whether or not they're applicable to humans. And then we'll go through what we've learned with our epidemiology colleagues about what the long-term effects of in utero SSRI exposure actually may be. So visiting the clinical question first, depression and pregnancy. Obviously, it's a big problem. 20 to, somewhere around 20% of women will exhibit signs of anxiety or depression during the pregnancy. Some are in the mild to moderate range, some are in the moderate to severe. The treatment modalities, if it's on the more moderate and mild side, people suggest psychotherapy. Sometimes that's not available. For the moderate to severe, the recommendation is to use medication. But in all cases, the idea is that we should be quite vigilant and aggressive about recognizing and treating anxiety and depression, but especially depression during the period of pregnancy. The outcomes that we fear most are suicidality of the mother, development into a psychotic depression. But then there are more other sort of common things that occur in the context of depression, which could be problematic for the fetus, such as a lot of evidence suggests that mothers with depression are not as good about doing all of the things they should do regarding prenatal care and nutrition. There's evidence that the state of depression is increasing stress hormones, such as corticosterone, as well as inflammatory factors that may affect the fetal development. There's evidence that women with depression tend to smoke more, don't abstain from alcohol, and engage in other substances, probably as a form of self-treatment. And then there's the risk that postpartum depression is going to be a problem, because usually postpartum depression is preceded by antepartum depression. And so the question that when I was a resident was, the focus was really on what were the proximal risks posed by SSRI use on the fetus. And so there was numerous publications that examined this. And there was evidence that maybe there was some premature birth, maybe low for gestational age birth weights. There was a transient syndrome. It was called a abstinence or SSRI abstinence syndrome that involves increased rigidity, temporarily poor feeding. But those usually subsided in 48 hours and were not ever tied to any long-term problems with the child. There were a few more frightening side effects that were noted, including persistent pulmonary hypertension, but this turned out to be very rare. And then there was some midline septal defects that were recorded regarding the infant's heart, but those also turned out to be exceedingly rare. And the increase was so minor that these, I don't think, were problems that really should have kept us from treating a mother who is in need. And so this was the idea then, that if you think about the risks of untreated depression, you have all of these factors that are potentially negatively impacting the fetus. And I think the belief is that by leaving these untreated, we were also causing problems for the eventual offspring as the offspring matured. There's evidence that prepartum depression was involved, linked to increased internalizing and externalizing disorders, and increased risk of depression and anxiety as that child reached his adolescent years. And so the idea was, if we could reduce all of these negative factors, we should, in fact, improve outcomes and have children with fewer of these factors, such as internalizing and externalizing disorders, and ultimately having lower rates of depression and anxiety when they reached adolescence. But the problem is that those studies, to my knowledge, as a resident, were never done. The first half of this equation, clearly the treatment clearly benefits the mother, and to the extent that all of these other potential negative factors were mitigated, it should produce these outcomes. But to my knowledge, at that time, those studies were never done. The second half of the equation had never been sort of demonstrated. I mean, the evidence was quite clear. This is a figure taken from Myrna Weissman's paper in 2006. It was part of the STAR-D trial. And this is just one example, but the change in the child's diagnostic outcomes as a function of the maternal response. So you have a depressed mother. This is all now postpartum. And so we're looking at the behavioral outcomes of kids as a function of improving mother's depression. So this is how well they responded to an antidepressant. And as you can see here, the change in the rates of diagnosis of the children was quite clearly improved by improving mom's depression. And this can be seen in internalizing behaviors or externalizing symptoms. That as the mother gets better, the child gets better. There's been a number of studies looking at the effects of both antepartum and postpartum depression on outcomes. I highlighted here the ones looking at antepartum, and they vary, some showing a quite significant effect on the risk of offspring depression later in life, others not so much. Even recently, this was from Mary Phillips. As some of you may have attended her grand rounds, I guess it was now a couple months ago. But even at the level of brain function, if you will, here she's looking at the executive control network and the salience network and showing the coupling of the amygdala with the salience network is altered in the child as a function of the mother's depression during pregnancy. So there was plenty of evidence to think that we should be trying to reverse whatever's going on in the mother. And if SSRIs can do that, it would logically make sense that it should be helping the child. And I think as a consequence of this thinking, as more and more we got comfortable with this idea that SSRIs were safe and that we needed to be addressing this problem of depression and anxiety during pregnancy, you can see here the rates of pregnancy using SSRIs went up quite linearly during the last couple decades, and it's showing this leveling off, but I believe this trend is continued. This stopped here in 2005. The use of other medications is quite rare, such as tricyclics and monoamine oxidase inhibitors. The treatment of choice is really the SSRIs. So again, this is just coming back to this idea that if the antidepressants are improving all of these factors that are impacting the fetus, are we actually producing these outcomes? And as I said before, the benefit of the SSRI use in pregnancy, this is not really ever been looked at. This was something that sort of bothered me when I was a resident. I mean, all of this made sense, but usually in clinical trials, we try and demonstrate that what we're actually doing is making a benefit, all the benefits that we're ascribing to it. So this had been a lingering question in my mind, but when I was an assistant professor, freshly out of Renee's lab and starting my own lab, we started working with mice that lacked the serotonin transporter, and found some things which we found kind of confusing. And we called this, at that time, the 5-HTT is the 5-HT transporter. So the serotonin transporter, it's the site of action of the serotonin selective reuptake inhibitors. So if this is the transporter here on the axon terminal of a serotonergic neuron, you can see here you have 5-HT, which is being packaged into vesicles, which then gets released. You have various receptors here on the postsynaptic side. And then in the cell body, you have 5-HT1A autoreceptors, you have monoamine oxidase enzymes, which break down, and you have TPH, which synthesizes serotonin. So when you're blocking the serotonin transporter, at least transiently, you're getting more serotonin in the synapse. And presumably it's been thought that this has a lot to do with the therapeutic effects of these medications. And we know that in mice, as well as in people, that we can change behavior in the adult by giving chronic SSRIs. And we can demonstrate in mice reduction in anxiety-like behaviors and behaviors that we think are linked to depression-like behaviors. Usually they're related to reactivity to stress. But what we had found in our early studies with mice that lacked the gene for this receptor, so essentially what we were doing was creating mice that genetically they were lacking. Essentially, we were doing genetically what fluoxetine is doing pharmacologically or other SSRIs. And what we had found was that, to our surprise, that by knocking this gene out, rather than creating mice that looked like they were on an SSRI, they looked like, if anything, they were on an anti-Prozac. They had increased anxiety and depression-like behavior. They had cognitive deficits. And we didn't really, at that time, understand why we would get these divergent effects. And we thought about it for a while and did some experiments and came to the conclusion that the reason the knockout was having such different behavioral phenotypes than what we're seeing with the medication given in adulthood had to do with its early developmental effects. And so what we did was a series of experiments. So if you imagine this is time going on this axis, and you're showing something here, like with this red bar, this is the lifelong blockade of the transporter that's seen like in the knockouts. And what we saw, again, these are the types of tests that we can use to assess these various dimensions of behaviors that we think have relevance to anxiety and depression in mice. The exact tests are not so important. It's the direction of these arrows. So if you look at adult fluoxetine, shown here in the green, all of these dimensions are being affected in significant ways. And when we started trying to mimic this early effect of the knockout by giving fluoxetine early in life, between the 2nd and 11th postnatal day in life, what we found was, in fact, all of these other behaviors were going in the opposite direction. So these animals were showing more depression-like behavior, more anxiety-like behavior, more neophobia. And we were able to show that this had a critical period or sensitive period. Originally, we were looking at a longer period from P2 to P21, which produced all of these effects. We then divided that period up into 2 phases, P2 to P11, P12 to P21, and we're able to show that the entire effect was contained within this earlier period of P2 to P11. Interestingly, just moving it out from P21 to P41 also had absolutely zero effect looking out later in life on these abnormal behaviors. The other thing that was curious to us, and I'm not going to show data on this, is that these mice actually looked pretty normal. So we give them this brief exposure, we stop, they gain weight, they do everything that looks normal. They're indistinguishable from their saline treated counterparts until they reach adolescence. And that's really where these increased anxiety and depression-like behaviors begin, which we thought was very curious. We'll come back to that. Now, the other thing that we tried to convince ourselves of was we were using fluoxetine, as it turns out, just because it had a longer half-life in mice than any of the other ones. But we wanted to make sure that this wasn't just something, a one-off. Was there something different about fluoxetine? So again, just pay attention to the arrows. This is what I said we were looking at with when you give an adult mouse an antidepressant. This is what we see with the knockouts. Again, we replicated all of those behaviors by just taking a normal mouse and briefly giving it fluoxetine early in life. We were able to reproduce the same effect by using clomipramine, which, if you recall, is one of the tricyclics that has the highest selectivity for the serotonin transporter vis-a-vis the norepinephrine transporter. It's about 35, 40 fold more selective for the serotonin transporter. We did look at citalopram. That was a surprise because citalopram has a half-life of about an hour or two in a young mouse pup. Yet even giving it once a day for that 11-day period or 9-day period still produced some effects, not quite to the same robust effect as we saw with clomipramine and fluoxetine. But the interesting one here is dizipramine, which is closely related to clomipramine. They're both tricyclics. But again, if you recall your early psychopharmacology notes, dizipramine is sort of the mirror image of clomipramine insofar as it has about 100-fold selectivity for the norepinephrine transporter. It prefers to block norepinephrine and has very little effect on the serotonin transporter. And even though these are very closely related or sort of chemically, dizipramine produces absolutely no detectable effects on behavior. And then just to follow this idea that it was serotonin and not norepinephrine, we took a completely different class of compound. This is chlorgeline, which will block the monoamine oxidase A. And early in life, given in the same time period, it largely reproduces the same effects as what we see with fluoxetine. So we took away from this that the mechanism of action ends up being of critical importance. Anything that affects serotonin metabolism, serotonin levels in this developing brain seems to produce these long-lasting effects on behavior that emerge around adolescence. And once they emerge, we've never seen them go away. We've studied these mice until they're would be ready for the old age home for mice. And we've never seen these behaviors diminish in any way. So being a basic science laboratory, we really wanted to try and understand what was happening in the brain that could be behind or underlying these altered behaviors. And so thinking about how to track this down and think about this in a logical way, we thought about presynaptic versus postsynaptic mechanisms as a way to approach this. So one thing we could look at is on the presynaptic side is serotonergic cell number. We could look at serotonin levels. We could look at how those neurons fired. Was there anything different about their firing rates? We could look at the fibers, like did they ramify, arborize, innervate other brain structures to the same extent. We could look at their terminals. And then we could also look at postsynaptic structures, the places that we thought could be playing a role in the altered behaviors that we were seeing in these mice. And we focused on three main areas: the hippocampus, the prefrontal cortex, and the amygdala. And the dorsal raphe really represents, as I said, these more presynaptic mechanisms. So in terms of effect on cell number, one of the reasons we were interested in this was that if you look at the raphe nucleus of an adult mouse and stain it for tryptophan hydroxylase, this was done in collaboration with Vicky Arango back in the day and Mark Underwood, you can see you have quite a few cells that stain positive for tryptophan hydroxylase, indicating that they are serotonergic neurons. If you looked in the serotonin transporter and knockout mice, the rate or the number of these neurons was greatly reduced, essentially by half. So if this is the raphe cell counts, the knockout mice were severely deficient in the number of neurons that had a serotonergic phenotype. Interestingly, this was not affected by this postnatal fluoxetine treatment, and that gave us some clues as to what might be going on. I mentioned firing. So one of the things that we had seen in the knockout mice is that their neurons, not only was there fewer of them, but they also fired at a much lower rate. Here I'm showing the firing rate in hertz of a wild type mouse. We happen to have a heterozygote mouse, which has one good copy, one bad copy, and it lies somewhere in between the wild types. And then in postnatal fluoxetine mice, so again, this brief exposure early in life to fluoxetine, it stops, you let the mouse grow up, and the firing rates of these neurons appear to be depressed forever. The question was why? Was this something intrinsic to the serotonergic neurons, or was it something like a circuit-based effect? And so we were able to use genetic techniques to stain these neurons green with green fluorescent protein, which allowed us to do slice physiology with Cheryl Beck. And what she found was that the electrical properties of these neurons were unaffected by the postnatal fluoxetine treatment, meaning that their intrinsic electrical properties were not altered. But what she found was that these neurons had a much higher rate of inhibitory drive from interneurons in the postnatal fluoxetine treated mice as opposed to the vehicle treated mice. And so this really does point to the fact that we are altering a circuit-based mechanism in terms of why these neurons are firing less. The other thing that we looked at was arborization of these neurons. So normally you have serotonergic neurons in the raphe. All of the cell bodies are quite concentrated, but those neurons will project and arborize and innervate almost the entire neuroaxis. And so this arborization, we wanted to find an independent way of examining this. And again, we turned to this genetic strategy, which basically liberates green fluorescent protein inside these neurons. And what we found was that the arborization was drastically reduced no matter where we looked. Here, I'm just showing you infralimbic cortex here, but if we quantify it in prelimbic or the anterior cingulate, you can see that the reduction in innervation in the postnatal fluoxetine treated mice is reduced across the board. And this is, this is showing also in the limbic cortex. Again, the dark lines here that you're seeing, these are serotonergic neurons in the vehicle treated mice, prelimbic, infralimbic. And if you look here in the postnatal fluoxetine treated mice, even without quantification, you could see that the density of serotonin neurons innervating these structures is decreased. Now, what about the amygdala? This is an interesting structure. we saw reduced innervation in the amygdala. This is comparing to again, the postnatal vehicle-treated mice. And we were interested in this because of the role of the amygdala in fear conditioning. And indeed, what we saw was that if you did context fear conditioning, and again, remember, this is where you put the animal, the mouse, in a certain environment and administer a foot shock in that environment. They will associate that environment with that unpleasant foot shock. And so preconditioning, you can see that freezing is unchanged as a function of this postnatal fluoxetine treatment. But after they received a few of these foot shocks, both animals are starting to freeze more once they enter this environment that's been associated with this unpleasant, unconditioned stimulus. But what we found was that the postnatal fluoxetine treated mice are showing more of this fear-like behavior, this freezing to context than their vehicle-treated counterparts. You can also do this using a tone. And again, what we found was associating the tone with the shock. It led to increased freezing in the postnatal fluoxetine treated mice. Both of these are amygdala dependent, which is why we were focusing on that. Now, interestingly, we had a postdoc in the lab that had come from a laboratory that actually has been able to image mice in an MR, magnetic resonance imaging, looking at increases in activity. And they can do this in live awake mice. They train them to not be completely freaked out, if you will, by being head fixed and placed in the magnet. And what they do, they were able to expose these mice to predator odors, such as fox urine, which they find quite terrifying. And they were then able to compare the activation of various circuits as a function of the exposure to this predator odor. Indeed, the amygdala as a function of, this is in comparison to postnatal fluoxetine treated mice versus vehicle treated mice. You're seeing higher activation in the amygdala, especially the right amygdala, in these males and females exposed to fox urine odor. And and you can also see that the periaqueductal gray seems to be more activated in both of these. So this is consistent with what we saw in terms of amygdala activation. And then the other thing that we were interested in is this idea of fear extinction, because it's one thing to learn a fear, but it's just as important to be able to understand whether that threat that you've associated with either a tone or the context remains relevant. And for that, this idea of fear extinction, you should be able to unlearn that or relearn that that something associated with a bad outcome is not something which is still relevant. So we were focusing on the medial prefrontal cortex, which is responsible for this extinction. And we found a number of changes there. First of all, we saw that the neurons that are in these areas had reduced complexity in their dendritic trees. And this is quantitated here. So the mice that would receive fluoxetine early in life, they had far less complexity of their dendrites. Not sure exactly what that means. But the thing that we did in collaboration with Evelyn Lamb up at University of Toronto is she did slice physiology in these infralimbic layers 2 and 3. And to our surprise, because I don't think anybody expected this. What we saw is if you looked at the infralimbic cortex, the neurons from normal or the vehicle-treated mice were far more apt to fire in response to an input current, shown down here, than the mice that had received postnatal fluoxetine. And again, this is months removed from their exposure. This is something which has been altered in these mice, and it hasn't changed despite them growing up. However, in the prelimbic cortex, we saw the exact opposite insofar as the postnatal fluoxetine treated mice, those neurons appear to be far more sensitive and apt to fire in response to an input current than their vehicle treated counterparts. And so what's the significance of this? Well, it was known for a while, and this was work done by Greg Quirk down at, he was in Puerto Rico, and one of the pioneers in trying to work out the circuitry of extinction. And what he had determined was that the prefrontal cortex, you have the prelimbic and the infralimbic cortices that have to coordinate. The prelimbic cortices need to turn themselves down in terms of their inputs to the basolateral amygdala, and they then impact on these central neurons in terms of affecting fear. Meanwhile, the infralimbic cortex needs to turn itself up and work with these intercalated cells in order to, and it's together that by interacting with these output neurons that you actually can change the response to learn fear and modulate it in such a way that you don't have to always respond in a stereotyped way. So looking at this again, so this is a tone-based, based fear conditioning. So these animals have been conditioned to freeze in response to a tone because they've been paired with a tone and a foot shock. And now what we're saying is we're exposing them to the tone over and over again without the foot shock. And that information should allow those animals to understand that that association is no longer relevant and they don't have to freeze every time they hear the tone. And so a normal animal here shown in blue, this is the vehicle treated animals, extinguish this freezing behavior quite efficiently after several exposures to the unpaired tones. The the fluoxetine treated mice really have a very difficult time letting go. Once they've learned this fear, they're much less apt to let it go. And this persists. This is trying to show that you can eventually get them down to the same level, but if you then wait a couple of weeks and then retest them, you can see that the fluoxetine treated mice still are reacting more, freezing more than their vehicle treated counterparts. The other thing that we had observed was that if we had made these mice, if you will, anxious and depressed by this early life exposure to an SSRI, could we treat that anxiety and depression later in life with our standard treatments, such as fluoxetine? And so we had evidence that, in fact, these animals would not respond to fluoxetine. And this, Sarah Canetta picked up on this and demonstrated again that if you had an animal that was treated with postnatal fluoxetine, you actually see to be making them worse in this particular paradigm. This is a novelty suppressed feeding than you do if the animals were naive. So it was a concern to us that that if these animals were becoming more depressed, if you will, or anxious, that we were creating a brain that was essentially going to be unresponsive to our normal therapeutic approaches. So let me just summarize then where we are. I've taken you through quite a bit, but just to recall that this early life SSRI exposure in mice seems to have multiple effects on behaviors that we think are relevant to psychiatric disorders. They're more afraid of novel environments. They appear to exhibit reduced resilience to stressful situations. They have augmented fear learning, so they will take the same, say, shock tone pairing and respond with more fear. And not only that, then they have more difficulty extinguishing that fear, even after multiple trials of showing that that pairing is no longer salient. And I didn't show the data, but these mice also appear to do more poorly on spatial-based learning tasks like the Morris water maze. And as I highlighted earlier, these behaviors do not manifest until after puberty. In terms of what we think mechanistically could be going on, we did find multiple things that are happening to brain structure. You have, as I said, reduced firing of the dorsal and median raphe neurons. Those neurons are not arborizing or innervating their key target structures to the same degree. We've seen in non-serotonergic neurons an alteration both in the structure of those neurons in the prefrontal cortex, as well as changes in their physiology, their likelihood of firing in response to an input in such a way that seems to be badly set up to extinguish learned fear. And I didn't show this data, but there's also some curious wiring differences in terms of ipsilateral and contralateral projections. Suffice it to say that, as many of you know, the structures that project from one area of the brain to the other, they often have a mix of ipsilateral and contralateral fibers. Normally that's all sorted out in early development, and a lot of those ipsilateral fibers end up being suppressed. But we have preservation, it seems, of these ipsilateral fibers in the postnatal fluoxetine mice. It's very curious. So how then does this bring us back to connecting these abnormal behaviors and the developing brain? And we had two hypotheses among many, but the first one was that a lot of these abnormalities could be linked to this reduced innervation and firing rate. And we think that we can link the neophobia and augmented fear conditioning and impaired spatial learning with those sort of presynaptic mechanisms. And that may be related to, we think, overactivation of 5-HT1A and 1B receptors during this critical developmental period. And the second was that these wiring abnormalities that are affecting non-serotonergic neurons, like say, for example, in the prefrontal cortex, are resulting from a blockade of serotonin transporters that are expressed oddly only during a transient period during early brain development. Let me tell you what I'm talking about. So before you have, this is a picture of a mouse brain taken in coronal sections. In the adult, this is the raphe nucleus here in G and H. This is where you expect to find the expression of serotonin transporter genes. This is looking at mRNA, but using a technique that allows you to see where the gene was expressed during the whole period of development. You can see that the brain during early development, this is starting roughly in the equivalent of the mouse of second trimester and continuing through third trimester. You can see in the thalamus, you're getting very high levels of expression. You could see that in the areas of the prefrontal cortex, you're seeing high levels of expression. This is in A, you can see the prefrontal cortex. There's a CA3 region of the hippocampus. We know that this is present, like I said, from probably day 10 or 11. So it's about mid-gestation for a mouse, and then it disappears about 10 days postpartum, which, as you'll remember, is the period during which we're exposing these animals to fluoxetine. Now, we were able to test this idea of affecting these transiently expressed transporters using a special line of serotonin transporter genes that where they had the LOXP sites, so we could use this to manipulate and knock out the transporter only in specific brain regions. We did that only in the cortex using a Cree driver known as EMX1 Cree. Suffice it to say that we preserved expression of the transporter in the raphe. We preserved expression of the transporter in the thalamus. But as you can see in the wild type mice, this is this area of the prefrontal cortex. You can see these little blue dots. That's indication of expression. And it's completely lacking here in these mice that we've genetically engineered to suppress the expression of the transporter here. So these mice are essentially as if we could use postnatal fluoxetine specifically in the prefrontal cortex. And so what did we find? We found that we could reproduce this behavior of being unable to unlearn a learned fear. So these animals are terrible at extinction of fear learning. This is the wild type mice. These are the mice where we suppressed the transporter in the prefrontal cortex. We also found that one of our tests of stress responsiveness is something called shock escape. It's very similar to learned helplessness. And what we found is that the mice that where we suppressed the transporter expression in the prefrontal cortex during development had a much higher latency to escape a shock. They have an open door, they can run out. Normally these mice will zip out very quickly. The transporter mice or the postnatal fluoxetine mice tend to not exit when they can. And this all goes back to this circuitry that we talked about before. So we think by affecting the medial prefrontal cortex, we're affecting learned helplessness, and we're affecting this extinction of fear learning that we've seen. So now the question we had was this expression that we see of the transporter in all of these bizarre regions of the brain transiently during development that only ever been shown in rodents. And so one of the things we did with our Conti Center was collaborated with primate researchers at University of California, Davis, and harvested macaques at different ages. And so here you can see in a second trimester macaque, we're focusing on this area here. This is part of the medial prefrontal cortex. you can see that the serotonin transporter gene is being expressed. This is mRNA in these regions. This is actually a higher degree of expression than what we see in rats or mice. And so you can conclude from this that this transient expression of the transporter in these bizarre brain regions is preserved in higher order primates, such as the rhesus macaque. It would be hard to imagine that we won't see this if we were ever to look at the human fetus. And you can also see here as the, just shortly after, this is a newborn macaque, this level of transporter expression is already starting to disappear. And by the time you reach an adult, it's completely gone. So again, this turns on during early brain development in utero, reaches a crescendo second, third trimester, and then quickly starts to revert back to what appears to be the adult form. So I'm going to try and speed up here, see if I can finish in the last next 5-10 minutes. So what about human fetal exposure? The one thing that I haven't emphasized is that the mouse is born quite premature relative to humans. And so the last trimester of mouse development, if you will, happens ex utero, which is the period of time where we were administering our fluoxetine. So essentially, this is the equivalent of the third trimester of human development. So it's relevant, we think, for what might be happening if mothers are taking SSRIs. Now, the one thing we know is that SSRIs taken by the mother readily enter the fetal circulation through the placenta. And so we were interested in trying to understand whether or not what we'd seen in mice has any relevance to what could be going on with the use of SSRIs in pregnancy. And this comes back to that original question I had, which is, we know that they're good for the mother, and we think we're improving the outcomes for the offspring through this treatment of the mother. It just stood to reason, but is it actually true? So to do this, we had to do these large population-based studies where we could link registers of maternal prescriptions being filled for SSRIs with pregnancy registers, and then finally linking those to the health outcomes of the offspring that were exposed. So we first did this in Finland, and what we saw was that we had several comparator groups. So we had one in black, which is the sort of gold standard moms that had no history of depression, no history of prior SSRI use, and no SSRI use during pregnancy. And you can see their rates of depression, anxiety disorders, autism-related disorders, or ADHD, were not zero, but lower, among the lowest. The second group we had was women with a history of depression, but no SSRI use prior or during the pregnancy, and those were the blue. So you can see they're higher because maybe the history of, the depression is having some sort of impact. So autism, ADHD, all are higher. And then you had mothers with a history of depression who were using SSRIs but stopped using them before pregnancy. These are the yellow, and they're somewhat in between, depending on the disorder. And then you have the women with a history of depression and using SSRIs both before and during their pregnancy, and that is this magenta line. And what we saw was, if you will, consistent with what we might have been predicting from the mice. The rates of depression are going up as these kids hit puberty. Anxiety disorders looks like they have an earlier onset. than the depression, which is something we also expected to see. But the rates of ADHD and autism really aren't affected by this maternal medication use, which again could be consistent with the idea that we didn't see any differences in the behavior of these mice until they hit puberty. We tried to control for as much as possible. We were especially worried that there was something different about the severity of the illness in the mother that continued taking these antidepressants during pregnancy versus those who stopped. But every attempt we made to adjust for either sex, covariates related to exposure and depression, we restricted them to just SSRI monotherapy, thinking that if women were using augmenting strategies, maybe they had a more severe depression. We adjusted for the number of episodes and for suicidal behavior. None of those really affected the analysis in any meaningful way. And then we were intrigued by a report that came out of Denmark a year later that essentially found the same thing. They were asking antidepressant use during pregnancy, and they were mainly just focusing on psychiatric disorders. So they had a similar design. They had an unexposed group, an antidepressant discontinuation group, a continuation group, and then a new user group. We didn't have that one. And if you're looking at the cumulative incidence of psychiatric disorders in the children, you can see the unexposed group again has the lowest rate. And the women who have a history of something that was requiring them to use an antidepressant are somewhere in the middle, but the new users and the continuation, women who continued through pregnancy, their children had the highest rates of psychiatric disorders. What's interesting is if you go to the supplemental data, you can break it down. They did look at mood disorders. These numbers are relatively low, but they did find that the rates of depression, even if it was only used, the SSRI was used for one trimester, doubled essentially. And if it was used for more than one trimester, went up by a factor of over 3. And so we had a grant with Adi Talati and our colleagues in Denmark. It was now 4 or 5 years later, and we were able to extend these findings and take a different approach. So we were, rather than doing a strict comparison with depressed and SSRI versus SSRI, we were trying to do a propensity analysis. And let me show you what I think was interesting. So the one thing, if you notice, the Finnish data stopped at age 14. And one of the things that we were curious about was what happens after that. And as you can see here, we have the unexposed. This is the Danish data. So the black is, we're looking at depressive disorders. The gray is anxiety disorders. They looked at adjustment disorders. And then interestingly, as a proxy, we can get into this. They were looking at what antidepressants were prescribed to these children as a proxy for psychiatric disorders that may not have been captured here. And so as you can see, unexposed versus exposed, we're seeing the same rates of these various disorders increasing before the age of 14. And these continue along probably what we would expect to be the trajectory they would as these kids mature in the unexposed. And the rates, though, in the exposed seem to be substantially higher. And let me break this down for you because this isn't the easiest graph to read. So if you look at the offspring outcomes at age 22, just sort of looking at the cumulative incidence of these disorders. So depression diagnoses are about doubled, anxiety diagnoses are more than doubled. And what's interesting is they looked at adjustment disorders. We didn't have that in our Finnish database. And And then the other proxy I was very curious about was whether or not these offspring of women who were either unexposed or exposed their children to SSRIs, the kids later in life are taking far more SSRI prescriptions than their unexposed counterparts. And again, you can see these are the hazard ratios. Let's jump over that. And then this is early work that we're doing with Jonathan Posner. This started back with our Conti Center, looking at whether or not the effects of SSRIs could be seen on newborn brain structure. The ends were very small, but they were able to find him and Claudia Lugo Candalas, changes in volume of the right amygdala and the right insula, as well as white matter tracks connecting those two structures. And the SSRI groups here are shown in green. The depressed, untreated, and the healthy controls look very similar in this study. No big differences, but the big difference does appear to be as a function of the SSRI exposure. So I'm going to summarize here, end it. Yeah, I think I can wrap up in the next minute. So just to summarize, I took you guys through a quick tour of what I've been up to. So I hope I've shown you that there's abundant evidence that supports this idea that 5-HT signaling early in life is influencing brain maturation. This is clearly true in the mice. And the interesting thing about the serotonin system, which is not often discussed, is that serotonin evolutionarily is one of the oldest and most conserved neurotransmitter systems. Whether you go from mollusks to man, almost any organism that has a semblance of a central nervous system uses serotonin. And the function of serotonin in these various brains seems to be relatively well conserved. So, which is one of the reasons we were thinking that what we found in mice may be applicable to what's going on in humans. And we think that this transient expression of the serotonin transporter in the fetal brain, it overlaps with our SSRI sensitive periods. We think this has mechanistic significance. And I think the outcome or upshot of this is that, at least in rodents, timing will matter immensely. It really will depend on when these SSRIs are being exposed, whether in terms of affecting raphe development versus these prefrontal cortical development. I didn't show you the data, but we also have shown that there's a dose response effect. So lowering the dose does appear to reduce the impact later in life. And the mechanism of action of these antidepressants matters. So not all antidepressants are created equal. It seems that it's really the ones that are affecting serotonin metabolism, either through reuptake or MAOAs, or, affecting the breakdown. Anything that touches, that doesn't affect norepinephrine, serotonin doesn't appear to have these effects. And this brings me back to the question I had, which is, are we helping or are we hurting these children who were exposed to these medications? I think it was the hope of everybody that all of these medications should be beneficial for the outcomes. We think that the data, there's still more work to be done, but there is a plausible reason to believe that what we see in mice could be happening in humans. And so far, I would say that what we've seen is that these medications, maybe they aren't hurting, but clearly they haven't had the beneficial effect that we had hoped for. So maybe I'm gonna stop there. We have some new studies that people can ask me about if they're interested. And again, just to give kudos where they're due, the incredible colleagues I've been working with for the last 20 years. So with that, let me stop and I can take any questions. Thank you.
Thank you, Jay, so much. That was wonderful. I loved it. I am going to start with some trainee questions, and then we can go from there. We have a trainee question from Hannah Simon. She is curious if any of the human cohort studies examine dose-dependent effects of psychiatric outcomes in the offspring exposed to SSRIs. And you had just mentioned dose, but can you give her some more information on your dose data?
Yeah, it's a really good question. We're trying to do two things with our colleagues in Denmark. One is exactly what you were saying, which is seeing if we can make some connection between the actual dosage and the outcome. And then the other thing that we're in the process of analyzing is the duration of exposure. So trimester 1, 2, or 3, or all 3, or just 2. We're trying to see whether there's any additive effects. But so far, I don't have a good answer for you, but it's a fantastic question. And I think it has relevance to how we can begin to think clinically about the way to best approach this question. Because if you can reduce dose or switch, which is harder, you may have some benefits down the line.
Thank you. We have another trainee question that I am going to combine with Alex Harris's question as well. We have a question asking about the use of non-serotonin depression meds such as Wellbutrin or stimulants. And Alex Harris also would like to know if you have thoughts on allopregnanolone would be a good choice of treatment for depression during pregnancy.
Yeah, it's two good questions. So did I stop? I guess I stopped sharing, right? Yeah. So the Wellbutrin question, we actually think we can answer that. Wellbutrin's not really used to any great extent in these Scandinavian countries, but we have a collaboration with the Mayo Clinic in Rochester, Minnesota. And they sort of, because of the intense participation of the surrounding community in their healthcare system and their electronic medical records, we can sort of create our own mini Scandinavia in that surrounding county. And they actually happen to have a very high rate of use of bupropion in pregnancy. And so we're in the process of examining whether the outcomes of bupropion exposed kids is similar or different than the ones who were SSRI exposed. Allopregnanolone, good question. I mean, I thought, yeah, I mean, for postpartum pregnancy, the one distinction I should make is that the rate at which SSRIs would get into a newborn via breast milk and the rate that it would pass through the placenta are hardly comparable, if that makes sense. The rate of the maternal plasma levels, you find about 70% of the maternal plasma levels in cord blood. So you get about a 70%, 80% transfer between the mother and the infant. Whereas in breast milk, you will find these medications in the breast milk, but they get into the fetus at such a low, or the newborn at such a low rate that it's essentially unmeasurable in the fetal or the newborn circulation. So if dose matters, I would be far less worried about exposure coming from breast milk than I would from in utero passage through the placenta. But yeah, anything that doesn't affect serotonin, I would think should be something we should be looking at.
Thank you. We have one more trainee question. They're asking, are there any studies which compare women taking SSRIs for something other than depression or anxiety? And the trainee is asking specifically for pain syndromes or bulimia with women taking them for depression or anxiety. They're asking essentially to control for SSRIs and assess depression, anxiety as a factor leading to negative outcomes in the offspring.
Yeah, God, another great question. So that it's harder to do than it sounds like. Again, I think it's really comes down to the way these large epidemiologic samples are done. We often don't have the kind of granular data as to, not all of them are diagnosed. It really is, it's a function of the way diagnoses get conferred in the Scandinavian countries. They're only a small number of the people who are taking SSRIs actually have a diagnosis. So that is a limitation of our study for sure. But to the extent that not everybody who's taking an SSRI or during pregnancy is taking it for depression, they could be taking it for anxiety, OCD, as you said, bulimia, pain syndromes, migraines. The percentage of those people relative to all SSRI users, it's hard, we just don't know. But if we could suss that out, I think that would be a useful way of trying to disambiguate the relative contribution of the SSRI versus maternal depression and all of the behavioral changes that come with it related to parenting. Both of those seem to be increasing the risk of these kids. And so that is something that we're trying to do in a study up in Sherbrooke, but that's ongoing. So I don't have a good answer for you.
Thank you. Dr. Lieberman is going to jump in and ask the next question.
Yeah, sorry for jumping in the queue, but has there been any effort to examine serotonin transporter genotype with the effects of this?
Yeah, there has been. And it's one study that I'm aware of, and the conclusion of that study was very small, was that the children with the lower expressing transporter variants were at higher risk than the higher expressing genotypes. So, was that what you were getting at, or am I?
Yeah, no, exactly. And I mean, the transporter has been kind of oversold as a genetic measure of guiding treatment or risk, but still it's an important gene to interrogate at least.
Mm-hmm.
But this is like a gotcha question, or putting you on the spot. So you've kind of released the genie from the bottle on this, and there's some responsibility incumbent in trying to solve it in terms of not just whether there is a risk, but what else can you do? The Betsy's question, or the question on Alex's question on allopregnanolone and what Sage Pharmaceuticals is developing is important to know. And there's a big story there, as you know, as well as others, with Steve Paul and Leslie Morrow and Bob Purdy about neurosteroids. So that's important to find out. The other thing is, if there is this selectivity of norepinephrine versus serotonin versus any other neurotransmitter, that you can dissect that. And you're probably old enough to remember the Shopson and Gershon famous study, where they showed by using alpha-methylparatyrosine and parachlorophenylalanine that it was the serotonergic component of the then combined SNRIs, the first generation, that was really the critical factor in sustaining therapeutic effect. Mm-hmm. Back then, there were some selective norepinephrine inhibitors, including nomifense, which have been withdrawn. But now there's some that have recently been approved as well. So in animals, you could do that AMPT and PCPA study to determine if there really is a selective neurochemical effect, and that would indicate that if you do have to treat people during pregnancy, what is the preferred pharmacology of the antidepressant?
Yeah. Good question. Yeah, I look. No, I think you can do these in animals. There's no way we're going to do a RCT of these various compounds. At least I don't foresee that.
Oh, but if you treated pregnant dams with a medication, an SNRI or an SRI, and you pre-treated half with PCPA, the other half with AMPT, and another with nothing, you could see if those behavioral the teratogenic effects, behavioral teratogenic effects are blocked or not.
Yeah, that it's a doable thing. I mean, I would be tempted just to like do something, a variant of what we did with dizipramine. I mean, dizipramine is relatively selective for norepinephrine reuptake, right? And we haven't done it, but we could do the same thing with bupropion. We could do the same thing with duloxetine. Is it duloxetine? I think is the relatively norepinephrine.
No, duloxetine is a triple uptake inhibitor.
Okay, so
that's Cymbalta. It gets dopamine too.
Okay, so forget that. So, but yeah, I mean, I think without doing the depletion studies, you could just depend on the relative pharmacology of these different agents is sort of the temptation I would have. But you would still want to know. That's why I'm excited about what we're doing at the Mayo Clinic. I mean, if we can see that Wellbutrin doesn't have the same outcome in the offspring as what we're seeing with SSRIs, I would. That's probably as close as we're going to get to having a clue as to whether the mechanism of action really is what we're seeing in mice is applicable to humans. But I know I see where you're going, Jeff. I mean, we can talk more about it.
Thank you very much. We have a ton of questions, so I'm going to try to get through a couple in the next 5 minutes. Meg Haney said, beautiful talk. This is off topic, but she wants to know, do you have any idea what prenatal cannabis exposure does to any of these outcomes that you've looked at?
We are looking at that. Claudia actually has a grant to look at prenatal cannabis exposure in the same cohort that we're studying up in Quebec, in the Sherbrooke catchment area. So maybe we should have Claudia give grand rounds in a year or two, because but that grant has been funded, and I think they will. They should have some answers in a year or two. But right now, I don't know.
Okay. We have a question from Jeff Miller. He also said, it's a fantastic talk. Do you think these compelling animal findings and retrospective human findings are enough to recommend changes to prescribing guidance for women with depression during pregnancy? What would you recommend to a family member with a significant depression treated with an SSRI who wants to become pregnant? Is there a way to ethically do a placebo-controlled trial of SSRIs during pregnancy?
Yeah, I don't know who would ever fund it. Is the big problem. But yeah, ethically, I don't know. I mean, it's a good. Yeah, I don't know. What would I recommend? I think the easier question is, what would I recommend? I think if somebody is planning a pregnancy, the safest thing would be to try and taper off their antidepressant to the degree that's possible. Try and sustain their emotional health through a good psychotherapy relationship. There's a number of studies, Meg Spinelli especially, among others, have shown that IPT and I also believe CBT are extremely effective during pregnancy. But right now, the recommendation is really only psychotherapy for mild to moderate, and it really depends on the severity. So I think, look, anytime you're looking at a medication that crosses the placenta and gets into the infant, and knowing that the medication may be affecting brain development, it's still a hypothetical, right? I think there's reasons to suspect that maybe these medications aren't as bad as we seem to be finding. The one thing that's been very hard for us to rule out, and I think it's a reasonable question, is whether or not there is something systematically different about the women who continue an SSRI during their pregnancy versus the ones who taper off or stop. And again, looking from 30,000 feet, like you're obligated to with these epidemiologic population-based studies, we don't have a good answer for that. We're trying to address that with this more granular study where we can more closely phenotype the mothers and the offspring in Quebec. But the one idea has been that that this SSRI use during pregnancy is a proxy for something else: severity of illness, family history, something else which we're not capturing, and that the SSRI may not be driving the outcome. It may be what the SSRI is representing in terms of the mother that's using it, if that makes any sense. So I would say if the woman is really, very severely ill, yeah, I think you shouldn't, I wouldn't deny her the use of SSRIs, because I think, I didn't get into it, but I do believe that we can find ways to treat the depression that arises if it's what is actually responsible for the increased depression that we're seeing in adolescents.
Thank you. So
Okay, I just ask one more question, Chris.
Sure.
Jay, do you know that the findings that you've published and your colleagues have been a deterrent? I remember when the black box came out in relation to Prozac to causing self-harmful behaviors. John Mann and Bob Gibbons wrote an editorial because they showed statistically that the rate of prescribing by doctors went down, and simultaneously or subsequently the rate of suicides went up. Do you know that yours has been a deterrent to practice?
I don't think it's changed practice yet. No. No.
Okay, we'll do one more question, and then I cut and pasted all the questions for you, so I can send them to you later if you're interested.
Okay, thank you. I'll do my best to answer them offline.
There was a question from an anonymous attendee that said, you mentioned that treating early fluoxetine exposed mice later in life suggested that they were resistant to treatment. Has anyone looked at interventions in juvenile mice to see if the emergence of the phenotype can be prevented after this SSRI exposure?
Simple answer is no. I was trying to think. We haven't found any way to avoid this yet, but we haven't exhausted all of the possibilities. The one thing that Sarah Canetta was able to show was that a antidepressant that's not used here, but is widely used in Europe, it's called tianeptine, is able to reverse the behavioral effects that we see in these mice, but it's a completely different mechanism of action. It has nothing to do with serotonin. So maybe that's a clue.
Okay, in the sake of time, I just want to thank you so much. We had a record number of people attending today, and it was an amazing talk, like always. So thank you again for enlightening us, and it's great to see you.
Yeah, thanks everybody. Thanks for your attention. And thanks, Christine, for being such a gracious host.
Have a good day. Bye, Jay.